Okay, so yeah, we're nearing the end, but I think we hopefully did a sort of our tour around spatial science. But to close the meeting, I thought that, this is a spatial biology project, so that the word is certainly we're not just focused on omics here. We tried to spread it around. I was looking for somebody who could just again kind of wrap it up and give us perspective on the field. So um, our next two speakers are going to be Barry Lee and Laura Shin. So I actually just found out that they both got some degrees at Johns Hopkins because they've known each other for a very long time. Jerry is now an associate professor of clinical medicine, molecular material science, and quantitative and computational biology at USC, and he's a CSO at Analysis Technology. And Laura had got her degrees from Johns Hopkins and the Austin University of Medicine and Science, and she now is professor of clinical surgery. The reason this talk came about is that it's actually asking Ron Fan, who many of us know in the spatial field, was my perspective, and he actually recommended Jerry. He's like, Jerry knows a lot of stuff about this. And it turns out that, yeah, in fact, Jerry's probably had a direct hand in a lot of the machines that we now operate on and that do spatial techniques. He spent many years at the NCI, at the Cancer Moonshot, and in that funding level. So don't correct me, Jerry, but I assume he's essentially the king of the POs, and he's been manipulating the direction of spatial biology for the last 10 years. But yeah, so he's gonna talk a bit about I won't explain, but they're gonna give a combo talk about the perspective of the field and how we can think about translation of spatial biology. So take it away.
Perfect. I know I'm in between, as Russ mentioned, you and happy hour. So I hope everybody can hear me okay. And it is a little late in the day, but real quickly, I really appreciate Alex and team for getting us up here. And quick disclosure statement, I will talk a little bit about my work as a consultant for the Department of Defense's Murtha Cancer Center. I do serve on the Scientific Advisory Board of Atlas Exomics. And certainly last but not least, I do serve as the Chief Science Officer for the Ellison Institute, a do-good for-profit that has spun out of USC. And one last caveat, I am no longer with the government. April of 2018 was my last day at the National Cancer Institute. And how many of you in the room know my deputy at the time, who now is actually the king, Sean Hanlon? And those of you that either have an HTAN grant, an HTAN submission, a BSOC grant, if you don't know Sean, please reach out to Sean, because a lot of the slides that you'll see from the .gov land generously provided by Sean. But just to give you a feel of my perspective and what I'm going to talk with you a little bit about, I'm a chemical engineer by training, and joined the National Cancer Institute in 2006, hired by then Deputy Director Anna Barker. I've always served in that role since then. Our job was to stay sort of a little bit ahead of the curve for all of the presidential nominees. For those of you that are not aware, National Cancer Institute is one of the only ones out of the NIH director that is a presidentially appointed position. So I had a different boss every couple of years, and they all wanted to do something fun. So all the different programs that you see there, including the Physical Sciences Oncology program, very close to my heart, as well as a small pilot referred to back then as the Human Cancer Genome Project, now the Cancer Genome Atlas, is something we had a real good hand in formulating and shaping. And then if you keep moving to the right, I know today is State of the Union Day. This is not a political discussion. The time that I spent with former Vice President Biden as his Chief of Technology and Research as part of the Cancer Moonshot really is his own experience. And fortunately, with his son, Beau Biden, we were just talking a little bit about GGM. The red arrow there is probably where I'm going to share most of my perspectives. Is one of the things that we came together and sort of pulled the community in to think a little bit about public-private partnerships when some of these single-cell, bulk, spatial omics stuff was sort of starting. Probably contrary to popular belief, we in the government do try to reach out and work together back then, and I still think now. And I think some of you that may not, again, know Sean, if I haven't said the word Sean enough, please make sure to reach out to Sean Hanlon. But the mission of this organization I joined in 2006 until today has always been to try to build transdisciplinary approaches, get them in, infrastructure standards, and be able to broadly deploy some data and get those tools and knowledge out really for patient benefit. And that has not changed. And I will say, talking with Sean a little bit of, what should we be trying to bring as a perspective here? I tend to look at this figure a lot because we're always the one that sort of tell people, FDA, NCI, and we don't care what you call your widget, right? You can call it spatial, single, CTCs, cell-free liquid biopsy. If it does not reproducibly and rigorously give you information, the name of the actual field is not hosing anyone, at least on our side. But again, I do feel like we're the sort of guy with the wheel saying, hey, please stop reinventing the wheel, because you're just going to run smack into some of the issues we've heard already throughout the day. But some of the things that I think, cancer research has certainly progressively changed and been redefined over the past 20 years. The part 2006 to 16 had a chance to see that these are cases where every single wedge on here is not a discovery biomarker. It is an actual biomarker that is used to stratify and or indicate a line of therapy. And if you jumped into cancer research today, I don't remember the person that was sitting here that just joined cancer research. Thank you for joining the fight. Lots has gone on. But if you think about what has changed in the past 20 years, just technology-wise, right? In 1997, we were told, for those of you that are old enough, know what AOL Instant Messenger is. Don't talk to strangers, right? If that person was born back then at 14, we are now asking strangers to deliver food to our house and or ride with them, right? Trying to explain to someone what is, something like a ChatGPT, or the fact that in 2000, our biggest accomplishment and most exciting technology was an 8 MB USB stick. That wouldn't fit any piece of my presentation. How can you talk with that community when on the flip side, right? What were we super jazzed about in 2000 as a biomedical field? We sequenced the human genome the first time. To my ONT colleague over there, yes, or our PacBio colleagues, not as complete as it was sort of announced April of 2022. But the other thing that I know very close to my heart is something that we started in 2006 as well, 18 years ago now, the Cancer Genome Atlas. And I think we have hopefully helped move that conversation along. How many of you guys use the Cancer Genome Atlas in one way or another? Okay. And I know you all hated it when it first started. It was really hard to use, hard to get data, heard it all. But all the things that we were trying to do, and everybody has their own interpretation of what TCGA is. And I always come back to this to remind everyone, in 2006, 18 years ago, what we were trying to do, we were trying to test the feasibility of large-scale genomic analysis technologies. But the parts that were very challenging for us, and I'm glad that we placed it in there, we got a lot of heat for it. We wanted to make the data publicly accessible before publication. We wanted to make sure that it also met the highest standards for protection and respect of research participants. I know everybody concluded their talks with thank you, but if you can think about 2006, what it was like to try to get publicly available genomic sequencing data out there. It was not the most fun consent form. And every Wednesday, the requirement was upload your data. Every Thursday, everybody would sit around, TCGA, and we'd point fingers at each other. It was the sequencer's fault. It was the pathologist's fault. It's the biobank's fault. It was the abstractor's fault. Why is the data not sort of reproducible enough? And I think those are some of the things that I would say I hear a little bit of that here and there still in this community, and that's fine. I think you just have to think a little bit about, what is the intent of your study? And the intent, again, of TCGA, those 8 years, $500 million spent, not just to make the 10,000 patient, 25 tumor type data sets available, but also the cases that didn't make it. We learned from every one of those cases how to appropriately obtain fresh frozen samples? How do you actually generate data in a large scale fashion? But we did it for that first time point in a patient's journey. Primary, untreated, late stage tumor. That's it. And when we were thinking about in 2011, 2012, during the ramp up of TCGA, where do we go from here? And these are old slides, you could tell because it's 4:3 instead of 16:9 widescreen. The two places we were thinking, and everybody kept asking, should we do time? Should we get the next sample from that next patient? How would we do that? If they had adjuvant therapy, would they all line up again like we did with TCGA? Should we go to the physics community in the PSOC program and ask for more deep dives into single cells, into every possible relationship? Certainly, we put our money where our mouth is a little bit and said, hey, let's, to understand biological heterogeneity, we funded a whole bunch of single cell technology in 2011-12. Just think a little bit about what kind of cell phone you were using then. And the debates that started immediately were those technologies got put right up against bulk technologies. So what you're seeing here is on the x-axis, sort of one cell all the way out to 1 million units per experiment. It doesn't have to be cells. These could be people too, if you're an epidemiologist. But why is it that if you run a 1 million cell experiment, bulk experiment 3 times, immunoblot, ELISA, that information is more valid, or even if you did 10 experiments to get that 3 million again of 100,000 cells like an MTT assay, why is that the right answer? And each one of these single cell technologies immediately got slammed with, hey, your technology, you can only measure 2 cells, 5 cells, 10 cells. Your technology just does not have the right precision and accuracy. And I think one of the things we were worried about is what happens if one of these technologies started to be able to measure 1 million events. And I do think there's many different devices over the years. This is an old slide, certainly showcasing Rong and Jim's platform back then. But now if you look at when these technologies started to be able to see 1 cell, 2 cell, 10 cell, was the next step to fund the NIH 2-cell program or the NIH 10-cell program. I think some of that we struggled a little bit. We didn't necessarily just jump to the word spatial, but how might you look at this and say, well, that might be the right answer of a subpopulation of your particular disease, whether that's cancer or not. And as I mentioned, what do? We went out and I really appreciate my co-chair back then, David Chang. We worked with the foundation of the NIH. It is not something, I don't know how many of you have worked with the foundation of the NIH. It is not part of the NIH. It is NIH's 501c3 nonprofit. But we went out and sort of asked these folks, not just academic, but government, highlighting Sean there again. But we also reached out to colleagues at FDA and NIST and said, well, how should we think about this space? How should we make this space robust? And I just wanted to show you a figure back in sort of 2016 when we were wondering, right, where is the readiness of some of these platforms? Codex, maybe Cytoff, GE Multiomics. How might we think about bringing them all together? And we ended up putting out a paper collectively and sort of said, hey, if you're thinking about a new technology, and ignore the fact that it, at the moment, it's a single cell analysis platforms, have you gone down that checklist? And I actually, throughout the day, have been adding brown arrows into things that I've heard throughout the presentations. Do you have any unusual sample requirements? I think we talked a little bit about that today. Do you have specific analytical verification validation study designs? Are you trying to jump too far ahead if the platform or analysis techniques aren't ready yet? How will you distinguish inherent biological heterogeneity, whether that's cancer or stem cell or neuroscience? And then certainly last but not least, and I give this a lot of credit if you guys don't work with Anne Plant, she's awesome. How do you think about developing control materials you can compare results to each other? And who's going to build that? Who's going to give you contrived samples that are reproducible? Those are all things I'm just going to throw out there. And for the sake of time, please feel free to come and find me. But I will tell you, this is still something that we struggled after working with industry and getting the public-private partnerships together to say, on one end of that patient journey, TCGA, the other end, things like NCI Match, AACR Genie, ASCAP Taper, Orion. I'll let you know what, you could probably tell what year this is. And I actually had to answer this question. I had to answer this question because Biden had that question for me like day one. He was like, I've been told by all these people that you did this TCGA thing. Could we do 1 million patients like we just did with the precision medicine or all of us cohort? And I said, no, sir, we cannot. He was like, what do you mean you can't? This is not enough money. You're not, what is that? And I said, unfortunately, in the United States, 85% of cancer patients are first seen, first diagnosed, first treated in a community cancer setting. How will you get to the 85 if TCGA for the 15, it took us 8 years to get 10,000 cases? How might we think about it? How do you think about a cancer center, a patient that might not go back to the same cancer center, and those opportunities where data is lost? And I think the part I got the most heat about, which is why I give Dr. Shriver and Dr. Hu a lot of credit for doing this work, it isn't just the pre-analytical variables of the platform. The amount of time it took for the two of them leading this effort to resolve 1,000+ clinical abnormalities in the TCGA clinical data set so that others could use it for context. Really excited that they were also part of some of the moonshot efforts that we did. I will not go through all 74 things that we've done. I will give you a quick snapshot of two. One that I think we've already talked about, sort of on one hand, the research recommendations, including the Human Tumor Atlas Network. The other public-private partnership that's very close to my heart, I was really appreciative that Colonel Shriver, the Cancer Center Director shown in the middle there, was willing to take the lessons learned from CPTAC. He was willing to take my round wheel instead of reinventing it, and said, let's do this and let's consent a longitudinal cohort of patients across the nation's only 2 national systems, Department of Defense healthcare system and Department of Veterans Affairs. And I will tell you, it is coming out shortly, our first version of TCGA2 with more longitudinal follow-up. One paper has been out for some time now on lung cancer patients. The red circle there is for Sean Hanlon. He is also our data repository lead for Apollo. So if you have any questions on how to access Apollo data, look at Apollo data. This also has some spatial components. Unlike TCGA, this is now serial section plus laser microdissection. It's not quite yet full spatial, but just a heads up. You haven't heard enough Sean Hanlon. These are, his resources again. Really appreciate him continuing the fight, thinking a little bit about how to aggregate both scales, right? There is no right scale, wrong scale. There's just, what can you tell me more about a patient and their journey? And the fact that the data is aggregated, publicly available as part of Cancer Moonshot 1 and 2 goals, very important. I do want to transition over to Laura, who I think reminds me and all of the physicians I get a chance to work with. We have all this awesome data, but the human cognitive capacity can take 5 facts and make a decision, whether that's your caregiver, your patient, your physician. How do you condense some of this information down so it is actually understandable? When somebody hears the word "you have cancer," they're not going to hear how cool your widget is. And then on top of that, I really appreciate that Alex really put us through a pace of different disease cases. You don't just have cancer. You might have other comorbidities, diabetes, hypertension, cardiovascular disease. You might have moved. How might we think about that? And then how might we think a little bit about the fact that every TCGA paper, the only author list that kept growing was the data analysts. And there's just not enough of them. And I'm not putting this in ChatGPT, getting some answer out that I could inform a patient. And if we're going to do that, how might we start? And the small thing that part of my USC duties, we actually had a very forward-thinking undergraduate that said, I want to learn TCGA, and I found all of my resources for them, every grantee that we've ever funded, and none of them had ever taught courses for undergraduates. And they have now, thankfully, a peer-to-peer network, a peer-to-peer teaching class. And if some of you are interested, all of them are looking for internship and or job opportunities. So happy to send them all to Russ. But this is the next generation, because this year is the first year in this September where an undergraduate is going to come into our universities post TCGA. They will know nothing else, but shouldn't all of this data be publicly available, high quality and curated? I hope so. Last but certainly not least, if you're not tired of hearing Sean, he also welcomes many of you. If you have not registered and signed up for this Atlas of Atlas Junior Investigator meeting, please feel free to do so. It does happen, unfortunately, in a few days. And with that, thank you so much for taking the time to listen. I'm going to punt this over to my colleague, Laura, surgeon, and who's going to take over and give you a little bit of, some of the other work that we've been doing outside of what I used to do.
So I've given this talk to Jerry's group before. There is a little bit of a trigger warning. There are some not great pictures. I'm Laura Shin. I'm a surgeon over at USC. And I actually study diabetic foot complications related to the bone. So I teamed up with Wrong actually to look and analyze some of the bone diseases that occur in diabetes. I'll flip through these slides pretty quickly. A large part of the reason why we're interested is by the time in most of our lifetime, we'll have patients with diabetes sort of in every facet. So everyone will have a family member, everyone will have a friend that has been affected by diabetes in some way, shape, or form. As a foot surgeon, these are commonly the diabetic deformities that we see on a regular basis. What happens is that these patients undergo these complicated disease processes that not only are they sick, but they also have these foot deformities that lead to these large deformities that actually lead to ulcerations. And scarily, every 2 minutes, a leg is actually amputated as a complication of diabetes. So when I talked to Jerry, we actually compared a lot of the diabetic bone diseases with cancer. These patients actually have mortality rates as high as 20% within the first 6 months. So looking back at this data, it's very sobering. We do everything that we can to save these limbs. We do everything we can to save these patients. But 50% of these amputations actually occur even on the other side. And 1 in 5 of these patients are dead within 5 years. So the mortality rate is pretty sobering. I recently got an NIH grant, my first NIH grant, actually studying Charcot disease. This is a largely underdiagnosed bone condition. So it's a discrete local inflammatory reaction. It causes a local osteoporotic reaction. It looks very similar to a bone tumor, whereas all the bones in the area around it are spared. But for surgeons, if you don't catch this early enough before you can offload it, patients walk all over it because of the neuropathy. They don't have sensation, and they develop these catastrophic deformities. So it's like walking on a broken ankle without having any pain for long periods of time. Unfortunately, there is no medical treatment. There's no medication we can give these patients. Oftentimes, they're misdiagnosed as having an infection, and so our options are relatively limited. And this is actually present in up to 30% of neuropathic patients. So they come into the hospital, they have this, they're not aware, they're not protected, and they're told, oh, this is an infection, we have to amputate right away. So a lot of people actually end up losing their legs before they're actually diagnosed. As a reconstructive surgeon, this is sort of some of the everyday things that we have to do. We have to figure out a way to basically take bone that's broken or to cut out a bone tumor or something. Like in the middle of the foot, we have to remove all of that and actually put 2 healthy ends together. And so these are the types of surgeries that we do. Unfortunately, a lot of our technology is limited clinically. And so this is my plea as a surgeon, and hopefully you'll get to reach out to a lot of your clinical colleagues. A lot of what we have sort of in our arsenal is pretty limited. Radiography can actually take 2 weeks. So these patients can show up into the emergency room. They don't realize what they have. And MRIs show local inflammation, but people really can't identify whether or not it's this disease process or if it's an infectious disease process. And surveillance can be really limiting or difficult, especially with the cost of MRIs and the cost of healthcare these days in general. So these patients are very remiss, or they don't want to go to the hospital over and over again to find out what's going on. So I showed this slide to Jerry and his group, and I said, what can I do? I'm a surgeon. I have difficulty actually putting these feet back together. And you can see, so this was sort of what I mapped out for Jerry to keep it very simple. you have this bone that's affected, and then you have this margin and you have this healthy bone. I have to put the two healthy pieces together. What can I do? And so we actually started to develop some protocols to sample this bone. And this is where I think a lot of the spatial technology would be really relevant, especially for us as surgeons, is to try to figure out what bone is healthy, what bone is actually sort of transitioning into this sort of Charcot state. And is there any way that we can prevent it? Is there any way that we can see if this bone is consolidated or healed overall? So we started developing some of this protocol for this fresh bone sample culture. And I'm happy to report we're actually one of the first labs that actually has a bone repository of Charcot bone. So we have tissue available for any scientist that's interested. And then we started looking at the verification of the different cell identity, and we started to see differences. And so I'll kind of skip ahead to looking at some of the technology that we're starting with. So Fakar here actually presented with one of his lightning talks, and we're starting to look at individual cells and how they interact with the environment. So is it just within the Charcot environment itself? Is it within the margins? And why is it only in neuropathic patients? So not all diabetic patients get this. We've had patients that have actually been post-chemo that get this. So after they've gone through all of their treatments, they develop this neuropathy and they actually develop this bone disease. So we're trying to figure out what that signaling system is, and are these stem cells or are these progenitor cells impaired in some way? Are they kind of programmed to say like, hey, you're going to go to an osteolytic capacity instead of an osteoblastic capacity? And so we're hoping that we can actually utilize a lot of this technology and bring it together. And so these are a few of the probes that we've been working on. We're also looking at identifying some of this osteolytic bone with probes that are linked to bisphosphonate therapy. And so these are some of the early animal models that we're using right now. And so hopefully in the next future, we'll actually have like a better characterization of this and at least predict. So perhaps there could be some sort of blood test or some sort of assay that we can say, hey, you have this bone disease. You're going to need to protect yourself. You're going to likely need surgery. Here are the things that we can do to kind of improve your mortality overall. And if there's any way that we can prevent amputation, that's what we're hoping to do. With that, I think I can bring us to the happy hour.
It's late. Are there any burning questions? All right. Well, if anybody has any questions for any of our speakers, please follow happy hour. And with that, I'll thank you all for coming to this second Cedar Sinai spatial biography. Thank you so much.